All right, so let's get started. We are on chapter 15, and this chapter is going to be split into two lectures. There's also a chapter, a worksheet, an activity. It's got a little bit of chapter 12 and a little bit of chapter 15 on it. I posted that on Canvas. Go ahead and also, once you've taken a stab at it, respond to my survey about any questions that you want me to go over, so that I can post a little video or some type of document going over any difficult problems before the assignment is due. So for today, for this chapter, the objectives are to know the mechanisms of protein sorting by signal sequences, to understand vesicular transport, to be familiar with secretory pathways used by the cell, and to understand how endocytosis imports proteins. So let's talk about protein sorting and signal sequences. So we know that proteins are made by ribosomes, but once they've been made, how does the cell know where to direct these proteins, right? Some proteins need to be functioning at the cell surface, some need to be in lysosomes, other in mitochondria, some in the cytoplasm, right? So again, how do proteins know where to go in the cell? And so every protein, when it's made, can have on the end of it a sorting signal. So this is a sequence of amino acids that aren't really important for the function of the protein, but are a code that tells the cell where this protein needs to end up. Right? So we have the sorting signal, and it is a sequence of amino acids on the end of a protein. So some things that we need to think about, right? How do you get a protein across a membrane? So if you want to get a protein into the mitochondria, that actually has two plasma membranes. Or if you want to get it into the Golgi apparatus, right? What is the mechanism to get a protein through there? So any protein that's going to be functioning either in the cytosol or in the lumen, right, inside of an organelle, is going to be water soluble, right, hydrophilic, but it has to pass through a membrane, this phospholipid bilayer that we've talked about as being hydrophobic. So there's certain mechanisms that the cell uses in order to help transport these hydrophilic large molecules across a hydrophobic plasma membrane. Now, there's three mechanisms that are used to transport proteins into organelles, and it depends upon the organelle that it's destined for, right? So if a protein needs to enter into the nucleus, it's going to use a unique mechanism called nuclear pores, which we'll talk about. If the protein has to end up into the majority of organelles, such as mitochondria, lysosomes, right? Those are going to be transported across the membrane, right? There's special proteins that help to carry that protein across the membrane. However, we have a third method. So this is going to be proteins that need to end up in the Golgi apparatus or that need to be exocytosed, so sent outside of the cell, proteins that need to be implemented at the cell membrane. Those are going to be transported by our third mechanism, which is vesicles. So the first thing, again, nuclear, if it's going to the nucleus, we have these nuclear pores. They are selective. They don't just let anything in. So only the correct proteins will enter into the nucleus. When a protein is getting transported across the membrane, say to enter into the mitochondria, it needs to be unfolded. So we know that as a protein is made, often it will start to fold up automatically due to hydrophobic forces. Now, as that once that protein's made and released, when it gets to the organelle that it needs to be inserted into, it's going to unfold again into a single chain so that it can more easily enter into that organelle. And the proteins that help with this are called translocators. And then lastly, again, proteins moving from one compartment to another. So often proteins that are going to be, say, sent to the lysosome, or like things that have been endocytosed and need to go to a certain organelle, things that are going to be secreted by the cell, um, things that are going to be working in the Golgi, they are going to be transported via vesicles. Let's talk about these first two routes. So if a protein is ending up in the nucleus or in things like the mitochondria or the peroxisome, it is going to be, or the endoplasmic reticulum, it is going to be directed using these signal sequences, all right? So signal sequences are not used for vesicular transport, right? Now here we have a table of some examples of signal sequences. I do not expect you to memorize any of these sequences, but it shows you, for example, what types of destinations these proteins have that are using signal sequences. And then you can also see where they are found. So some of these are found, for example, import into the ER. A protein that's going to end up in the endoplasmic reticulum will have a signal sequence on its N-terminal end. You can see in that example signal sequence that it starts out with H3N, right? That's the N-terminus. And there's certain patterns of hydrophobic and polar, non-polar amino acids that can be recognized by the translocators to know if this is the protein that should enter into this or that organelle. Others, for example, a protein that's going to end up in the lumen inside the endoplasmic reticulum has a specific pattern that is found at the C-terminus. Others can be found more in the middle of the protein, such as those that are getting imported into the nucleus. Right? So, how could you design an experiment to determine the signal sequence for a protein? Let's say you have a protein that you're interested in, and you know that it ends up in a certain organelle, and you know the sequence, the amino acid sequence, but you don't know what part of that sequence contains that signal peptide or that signal sequence. Think about how you could design an experiment to determine what part of that protein contains the signal sequence. So I encourage you, this is something that I would normally pause in class and have you work with a partner. Pause the video now, brainstorm some ideas that you can think of, right? So here is one. It's not the only experiment that you could design to determine the signal sequence for a protein. So in this example, we have your protein of interest is normally sent into the endoplasmic reticulum, so it functions in the ER. And we're going to propose that the theoretical signal sequence is shown in red. Okay. Now, we could take our proposed signal sequence and attach it to a different protein. So for example, we have a protein that we know has no signal sequence because it functions in the cytosol. So it's kind of interesting. If a protein's function is to, or if its location it needs to end up in is in the cytosol, it has no signal sequence. So what we could do is we could take the sequence that we propose is the ER targeting sequence and stick it onto a protein that normally would not go to the endoplasmic reticulum, and then track that protein and see where does it end up. So in this case, they've taken the sequence that they propose in red, and they've attached it to a cytosolic protein, and they've found that now the cytosolic protein ends up in the endoplasmic reticulum. So they've successfully been able to identify what amino acids are responsible for directing a protein to the endoplasmic reticulum. And oppositely, you can see that the protein that normally ends up in the endoplasmic reticulum is now found in the cytosol. So think about what would happen if you thought that the opposite end of that protein was the signal sequence, right? If you took that sequence and attached it to the cytosolic protein, what results would you get? So if you were wrong, what would you see? What would happen is that your protein that normally gets targeted to the endoplasmic reticulum would still end up there, And your protein that normally is targeted to the cytosol would stay in the cytosol, right? Now, how do they actually do this technique? I don't expect you to understand that. But again, if you think about it, DNA, RNA, protein, if you're trying to alter the sequence, the polypeptide sequence of a protein, you're going to be changing things at the DNA level. So let's talk about this first target destination, the nucleus. Here we have a cartoon of the nucleus, a cross section, and you can see that, interestingly, the nucleus is actually continuous with the ER, which is kind of cool. Also, we have these big pores, right? These nuclear pores, and that is how things can enter or leave the nucleus. Another thing that you should note is that just like the mitochondria, the nucleus has both an inner and an outer phospholipid bilayer. Here is another perspective of the cell membrane of the nucleus. So you can see we've got our inner and our outer nuclear membranes, and spanning both of those layers is our nuclear pores. These are pretty neat proteins or complexes. They've got side chains that extend into the cavity of that pore. That creates kind of a gelatinous consistency. We also have on one side a nuclear basket that kind of catches things as they are coming through. And then on the opposite side, we have cytosolic fibrils that help guide the protein, if it's the correct one, into the pore. So we have some electron micrograph examples in B. You can see that cross section, and we can easily see the nuclear pore complexes and even the basket kind of hanging down below. And in C, we're looking from the outside of the cell, down of the nucleus, down onto the surface of the nucleus, and each of those little flower-shaped structures is a pore. It's pretty cool. So again, we've got these portions of proteins, that these sequences that are extending into the kind of channel area, the gap in that pore, and they are forming a mesh-like substance. They have, I said they kind of have a gel, they create a gel-like consistency, and that is due to the fact that these unstructured regions of proteins, these loops and things like that, that are sticking out into the pore, are all just containing repeated amino acid sequences. So a region of protein that would be extending out, for example, could just be a bunch of alanines. Right? And you don't need to know why, but when you have a ton of these repeated sequences, it tends to promote a gel-like consistency. And what this does is this prevents really large molecules from getting through and helps to promote the selectivity of the nuclear pore. Again, we're letting small water-soluble molecules into the nucleus using these nuclear pores. So let's brainstorm for a minute. This is another question that I would pause in class to ask you. What molecules need to cross the nuclear membrane? So pause the video, take a minute, and brainstorm what types of things would need to either enter or exit the nucleus. So the first thing that we would think of, right, would be RNA. Message RNA needs to leave the nucleus in order to be made into protein. We also have certain proteins whose function is in the nucleus. So for example, transcription and transcription and DNA replication both occur in the nucleus, and all of those require a host of proteins in order for those processes to occur. So proteins that get made out in the cytosol need to enter into the nucleus. And so proteins that are going to be targeted to enter the nucleus have a nuclear localization signal. So a certain signal, a sequence of amino acids on that protein that when that protein reaches the nuclear pore sees that sequence and says, okay, you're allowed in. We also have cytosolic nuclear import receptors. So these are going to aid with the transport of this protein across the nuclear pore. And so you can see that here in the figure in blue. It looks like an S. Right, so these nuclear import receptors are proteins. They're just hanging out near the surface of the nucleus. And they're going to check out prospective proteins that may need to cross the membrane. So they will read the localization signal if it has one, and if it's the correct signal, the nuclear import receptor will bind to that protein that needs to get into the nucleus. And the cool thing about this is that it acts kind of like a chaperone. It takes that protein and guides it through the nuclear pore, and it helps to disrupt that gel that is in the core of that channel, of that pore, and allow that protein to get through onto the inside of the nucleus. And once it has been transported through, the nuclear import receptor will detach from that protein. Let's talk about proteins that end up in other organelles, such as mitochondria or chloroplasts in the case of photosynthetic organisms. Again, we have in mitochondria, we have a double membrane. But again, we have a protein that has a signal sequence in red shown in this cartoon. And so on the surface of the mitochondria or the chloroplast is a protein called a translocator. So it's important to know that term. So this translocator has a region of it that will identify the signal sequence in a protein and determine whether it's the correct signal sequence for this destination. So if it reads the signal sequence and that protein is the correct protein that needs to enter into the mitochondria, the translocator will help to kind of unfold that protein into a long chain and thread that protein through the phospholipid bilayer. So it's guiding it through. And then there is another translocator on the inner membrane that will do the same thing. It will thread that protein through. Once that protein is into the matrix of the mitochondria, it can refold, and that signal peptide, that signal sequence isn't needed anymore, and so it gets cleaved off. And now this protein that originally was considered a precursor because it wasn't in the right place yet and it had a signal sequence on it, now is considered a mature protein. Peroxisomes are a unique situation, all right? So proteins can enter the peroxisomes from both the cytosol or the endoplasmic reticulum. So you can have ribosomes that are floating in the cytosol and also that are on the surface of the endoplasmic reticulum. And so there's proteins that are needed in peroxisomes from both locations. Before we get into that, let's talk about, first of all, what are peroxisomes? Because we don't talk about peroxisomes very often in our class, right? So some of the main functions of peroxisomes are to break down certain molecules such as toxins, fatty acids, and they also can make phospholipids. So if you have a protein that's made in the cytosol that needs to get into the peroxisome, it's going to be guided by receptor proteins. Again, this protein translocator. So just like with proteins that are entering into mitochondria or chloroplasts from the cytosol, proteins from the cytosol that need to get into peroxisomes are going to use signal peptides, signal sequences, which will tell a protein translocator to take that protein and thread it through the plasma membrane into the peroxisome. Sorry, made a little mistake. So in this case, it's a little bit different. Even though it's using a translocator, the proteins actually don't have to be unfolded. Now, proteins that are going to enter the ER, or to enter the peroxisome from the endoplasmic reticulum, are going to arrive via vesicles. In general, anything that leaves the endoplasmic reticulum is going to be leaving packaged as a vesicle. Right? So that's an important thing to know. So if a protein is made in the endoplasmic reticulum, it's going to bud off of the endoplasmic reticulum in a vesicle, and then that vesicle can fuse with the peroxisome and release its contents into the peroxisome. Alternatively, it's kind of interesting, that vesicle can be turned into a new peroxisome. So whatever other proteins are needed to create a peroxisome will be imported into that vesicle until eventually it has everything it needs to become a peroxisome. And this can happen, a similar method happens for making lysosomes as well. So here we have a picture of the endoplasmic reticulum. So in this experiment, scientists injected a dye into the interior of the ER that is a fluorescent green. And so you can see really how complex and networked the endoplasmic reticulum is. All right. So things that leave the endoplasmic reticulum are always going to leave via vesicles, right? So the endoplasmic reticulum is an entry point for proteins that are destined to other organelles. Once inside, these proteins do not re-enter the cytosol. So if a protein has a function in the cytosol, it never is going to enter the endoplasmic reticulum. Right? It's going to be made not by ribosomes on the surface of the ER, but by ribosomes that are free-floating in the cytosol. And again, these proteins, they are going to be made by ribosomes on the surface of the ER, right? So as soon as they're made, they're getting inserted into the ER. So any protein that gets inserted or made at the surface of the ER is only going to leave the ER as a vesicle, right? So this is different than our signal peptide sequence mechanism of travel. What is a vesicle? Again, it's just a little like lipid droplet, right? It's a phospholipid bilayer, and the interior of that vesicle is hydrophilic, right? So two kinds of proteins are transferred from the ribosome to the ER. First, we have water-soluble proteins. So proteins that are going to have a lot of polar side chains, right? They are hydrophilic. They tend to function inside of organelles, right? They need to be translocated across the membrane of whatever target organelle they are entering, and they need some help to do that because that membrane is nonpolar. Alternatively, they could also be secreted by the cell, right? So water-soluble proteins that are made at the rough ER are going to either be functioning inside of an organelle or they could be secreted by the cell itself. We also have transmembrane proteins that are made by ribosomes on the surface of the ER. So these could be proteins that have a function on the surface of the cell or in the plasma membrane of any particular organelle. So these get only partially translocated across the membrane. Right? So the ribosome makes the protein. It has a signal sequence that tells it that it needs to go to the ER, and it only gets put in halfway into the ER. It gets kind of stuck in the membrane. So again, this is useful if that protein has to have its function in the membrane, either on the surface of the cell or in the membrane of an organelle. Now, how do proteins get into the endoplasmic reticulum? Again, these are going to be proteins that are made by ribosomes that are on the surface of the rough ER. And as they are being made, they are being inserted into the ER. Right? So this is different than proteins that enter the nucleus or the mitochondria, right? They are fully formed proteins that travel to, say, the mitochondria, then they have to unfold and enter, right? So again, how is this possible? The ribosomes that are making that protein that ends up in the ER or literally on the surface of the endoplasmic reticulum. And here we have a picture of the rough ER where you can see discrete little black spots. Each of those are ribosomes, and they're on the surface of the ER. You can also see how the ER is very complex, right? There's lots of little tubules, and these proteins are getting inserted as they're made into the ER. So again, here are two different possible destinations for proteins that are made by ribosomes on the surface of the ER. On the left, if you look closely, on the surface of the rough ER, you can see these kind of orangish brown spots. Those are the ribosomes coating the ER. Right? So proteins are being made by these ribosomes, and as they are being synthesized, they're directly inserting into the endoplasmic reticulum. And when they need to, say, be secreted by the cell, they are going to bud off of the endoplasmic reticulum into a little vesicle, and they're going to travel through the Golgi and eventually to the surface of the cell, and you can see at the plasma membrane those proteins are being secreted out into the cell surroundings. We also talked about proteins that have functions inside, say, the plasma membrane, right? Say on the surface of the cell. And so that's the example that we have on the right. We have ribosomes on the rough ER that are synthesizing proteins, but these proteins, rather than completely entering into the lumen of the ER, they get stuck partways in the membrane of the ER. And so what happens when the ER buds off, that vesicle is going to have in its wall, in its phospholipid bilayer, that protein. Again, it's going to travel through the Golgi apparatus, and that vesicle is going to fuse with the cell surface, the plasma membrane on the cell surface. And when it fuses, now we have that protein already inserted into the plasma membrane. It's pretty cool. So it's not like the protein has to travel to the cell surface and then get inserted into the plasma membrane. It gets inserted into the plasma membrane as soon as that phospholipid bilayer is made, or as soon as that protein is made way back in the ER. And if you remember last week, we talked about how does new cell membrane get formed? It comes from the endoplasmic reticulum. So not only are more phospholipids being brought out to the cell surface, but encased in that phospholipid bilayer are proteins that are going to be needed at the surface of the cell, which is pretty neat. A little more detail about ribosomes. So how does a ribosome know whether it's supposed to be on the surface of the endoplasmic reticulum or in the cytosol? How does the protein go to the right ribosome, right? How does the protein make sure that it gets made by a ribosome on the surface of the endoplasmic reticulum versus in the cytosol? So interestingly, ribosomes, whether they are found in the cytosol or on the surface of the endoplasmic reticulum, are identical, and they can actually move about the cell, sometimes be attached to the ER and sometimes be free-floating. And so when a ribosome interacts with a message RNA, the message RNA really tells the ribosome where to go. So if a ribosome is in the cytosol and it interacts with message RNA that says it needs to be inserted into the ER, that ribosome plus its message RNA will travel to the ER before the rest of that protein gets made. And so that's what you can see in this picture, right? On the left, we've got a blue message RNA that has no signal sequence. So when it interacts with that ribosome, the ribosome doesn't need to go anywhere, and it can start making that protein. However, on the lower left, you can see that there's a message RNA, and that as that protein is starting to be made by the ribosome, it makes the signal sequence that tells that protein that it needs to go to the ER. And so the ribosome says, oh, wait a minute, we are in the wrong spot. It pauses making that protein. The ribosome travels with its message RNA to the endoplasmic reticulum and kind of docks at the surface of the ER. And once it's done that, only then will it continue to make that protein. Okay. Another thing that this figure demonstrates is that you can have the same message RNA be synthesized into protein multiple times, right? You can have multiple ribosomes traveling along a single message RNA strand, which is pretty cool. Let's get into more detail about how these translocators work, all right? So we have a ribosome. It has received a message RNA. It started synthesizing the protein based on that message RNA template, and it synthesized a signal sequence for the endoplasmic reticulum. So it paused making the protein, and it travels to the endoplasmic reticulum. Now, the way that it docks with that endoplasmic reticulum is a protein called a signal recognition particle, or SRP, right? So you should know the steps of this, right? So the signal recognition particle will only bind to proteins that have the ER signal sequence. So in this case, the signal recognition particle reads the signal sequence on this newly synthesized or in process of being synthesized protein. It says, yep, you're supposed to be here. And it recruits the whole complex, the ribosome, the messenger RNA, the new protein, everything, to the ER by binding to an SRP receptor, right? Signal recognition particle receptor. So now we have the ribosome in close contact to the endoplasmic reticulum. Then it's going to, that SRP receptor is going to guide the new protein into that translocator, right? So the protein translocator now has the protein as it's being synthesized is traveling through it. And now the ribosome gets a signal that it's okay to continue making more of this protein. Again, SRP protein binds the ribosome and the correct signal. So if it sees there's an ER signal on the newly forming polypeptide, it will bind to that. And it will slow down translation. Right? The ribosome takes a minute, pauses, and the SRP is recognized by the SRP receptor on the surface of the endoplasmic reticulum. That SRP receptor guides the ribosome to the translocator and helps to insert protein into the ER as it's being made. For some reason, I can't go back on this presentation method, so sorry. I'll try to be more careful. So if you have a protein that's going to end up inside the lumen of the ER, inside the cavity of the ER, this is a cartoon that demonstrates what happens next. So this protein, this newly forming protein has been inserted into the translocator due to the aid of the SRP receptor and the signal sequence. Right? And so now as that polypeptide chain is being synthesized, it is being threaded through. Right. Now, an important thing to note, the signal sequence for proteins that are ending up in the ER is on the N-terminal end of the protein. Right? It's important to note that. And so as that protein gets threaded through, it's a little backwards from what you would expect, right? The N-terminal portion of that protein is facing the cytosol. And then the C-terminal end kind of gets pushed through. So once the rest of that protein gets pushed through, we have a signal peptidase, right? The protein doesn't need that signal sequence anymore once it's been made and it's in the right spot. So this peptidase, it's ACE, an enzyme, it's cleaving peptides. It cuts off that signal sequence, and that signal sequence kind of floats away. And that releases the protein into the lumen, and it can refold and now have its function as a mature protein. Now, say you have a protein that needs to be inserted into the plasma membrane. Say it has a function on the surface of the cell. So it's going to have, in addition to the signal sequence, it's going to have a region called a stop transfer sequence. So both the start transfer sequence and the stop transfer sequence are hydrophobic, and that allows them to be in the plasma membrane. So the start transfer sequence is basically the same in most cases as the signal peptide. Right? So again, the N-terminal end of this protein is going to insert into the translocator, and it's going to be facing with the N-terminus out, facing the cytosol. And again, that protein is going to be threaded through the translocator as it is made. However, when the stop transfer sequence, that hydrophobic sequence, enters the translocator reads that and it stops transporting any more of the protein into the lumen of the ER. It recruits that signal peptidase. The signal peptidase cuts off the signal peptide or the start transfer sequence, and then the translocator leaves, and now we have a protein that's only been partially translocated across the membrane, and this is a transmembrane protein, considered in this case a single pass transmembrane protein. So if you see a hydrophobic region stop transfer sequence, you know that this protein is going to be inserted into the phospholipid bilayer. So let's think back about secondary structures. Right, when we talked about proteins in chapter 4, what type of structure would you expect to span the membrane? Right? We talked about alpha helices and beta pleated sheets and some of their potential functions. And we talked about how alpha helices tend to be the secondary structure that is found in regions of proteins that are inserted into plasma membranes. So again, this hydrophobic stop transfer sequence likely contains an alpha helical structure. And again, all those side chains on that alpha helix are non-polar so that it can sit in that phospholipid bilayer. Let's look at another kind of transmembrane protein, a double pass membrane. Right? So in this case, the start transfer sequence is further into the polypeptide chain. So it's called an internal ER sequence. Right? So this protein is getting made by a ribosome, say it happens to be out in the cytosol. As soon as that start transfer sequence gets made, it's also functioning as the signal sequence. The protein pauses, the ribosome pauses protein synthesis, moves to the ER. This sequence is recognized by the SRP. The SRP receptor recruits this whole protein to the translocator, and now it gets inserted into the translocator. And again, when this protein gets inserted, you always have the N-terminus facing out into the cytosol, right? And then the protein, the translocator allows protein synthesis to continue to occur as it gets threaded through. If that translocator identifies a stop transfer sequence, it will pause and it will let go of that protein. And so now what we have in this case is a protein that is a double pass transmembrane protein. It passes through the membrane twice, and both the N-terminus and the C-terminus are facing out into the cytosol. So it's pretty interesting. There are actually some proteins that pass the membrane 7 times, and we'll talk about those proteins in a future chapter. So let's do a problem here, right? This is something that you should be able to solve. I have a cartoon of a protein, and we know that it normally resides in the plasma membrane. Boxes 1 and 2 represent membrane spanning sequences. So those are hydrophobic sequences of the protein, and the arrow indicates where the signal peptidase is going to cleave. So we can predict how this protein is going to orient itself in the plasma membrane. So you have some different options that you can choose from here. So take a minute, pause the video, think about how this would get inserted into the translocator, what would happen once protein synthesis continues, and what would this protein end up finally looking like in the plasma membrane. So let's work on this problem in more detail. We know that that arrow indicates where a signal peptidase is going to act. And so that tells us, hey, if it's cutting there, that region 1 is probably the signal sequence, right? Because it's going to get cut off. So we can correspond, again, that region in this cartoon as the signal peptidase site, where that peptidase is going to cut, and that this red region is corresponding in our cartoon to the region 1. And we also have a region later on in the protein that is probably a stop transfer sequence. So this protein, as it's going to get inserted into the translocator, we know that the N-terminus region is going to be facing what? The cytosol. Right? And then the rest of that protein is going to be kind of shoved through the translocator until the translocator gets to that stop transfer sequence, which we have determined to be that region 2 in our cartoon. Once it reaches that region, the translocator sees that, it pauses, and it recruits that signal peptidase. The signal peptidase is going to cut at that site we indicated with the arrow and release what end into the lumen? The N-terminal end. N-terminal side of that protein is facing into the lumen of the ER. It has now the translocator is going to let go. And so what we have resulting is a single pass transmembrane protein with the N terminus in the lumen and the C terminus facing out into the cytosol. So you got to keep track of those N and C termini. So we've spent a good amount of time talking about signal peptides and translocation, all right, the details, the nitty-gritty of how that works. Objective 2 is going to go into more detail of how vesicular transport works. So once a protein leaves the endoplasmic reticulum, how does that vesicle form? How does it go where it needs to go? Here, this cartoon nicely shows something called the endomembrane system. It's basically anywhere that vesicles are going, starting with the ER, or if they've been endocytosed, for example, right? So the endomembrane system is going to include the ER, the Golgi, vesicles that have been endocytosed or that are on their way to be exocytosed, and also the lysosome. An endosome basically is like a precursor to a lysosome. So you can see a protein that's going to end up outside of the cell. It gets made by the ER. It's encased in a vesicle that buds off of the ER and has to first go to the Golgi apparatus. It travels through the Golgi apparatus, buds off as a vesicle again, and fuses with the cell membrane on the surface of the cell and releases its contents out into the extracellular environment. Conversely, the cell can take up proteins or other particles from the outside of the cell. It will form a vesicle again, budding into the cell, and that vesicle will travel either directly to the lysosome or via an endosome. And then that vesicle gets broken down, and the lysosome and the endosome kind of decide what to do with those components. So the ER is often only the first step in protein sorting, right? After the ER, unless that protein has a function specifically in the endoplasmic reticulum, it is always going to go next to the Golgi apparatus. So you should know first the ER, next the Golgi apparatus. And so what the Golgi apparatus does is it helps to further modify these proteins, put adjustments, modifications onto them, and also to help sort out where this cargo needs to go. Right, so how do these vesicles know where to go? How do they end up at the correct organelle? So let's talk first about how this vesicle gets made, right? So there is something called a protein coat on the surface of a vesicle. when it's being made, and that helps to form the shape of this bud-like structure. And this protein coat also helps to determine what gets put into that vesicle. Here we have an electron micrograph. You can see a vesicle forming. And on the right side, you can see that there's all sorts of these little black spots that are helping to form that round type bud shape. And they are also helping to recruit on the left side of that cell membrane the contents of the vesicle. And so you can see now the fully formed vesicle with the protein coat on the outside and a specific cargo inside of that vesicle. And then we also have a picture that shows just kind of from the outside what these vesicles look like. And you can see all these little bumps on them that are the proteins that are on that protein coat on the outside of the vesicle. All right, sorry about that awkward moment. My computer decided to freeze. But let's move on. So let's talk more about that protein coat. So one of the most commonly described protein coats is made out of clathrin. So you should know about clathrin. And in this cartoon, it's shown in green, and it has kind of a curved umbrella-like shape. And so you can see that because of this shape is kind of curved of the clathrin, it's actually going to physically force the plasma membrane into kind of a spherical type shape. So it's not just the cartoon, like these proteins, these clathrin proteins really do have this curved structure, which I think is really neat. And so as they associate with the plasma membrane, they promote this budding vesicle shape. And then we said that they also, these proteins that coat the vesicle also determine what actually gets put into that vesicle. So you can see that the protein clathrin is interacting with the cargo via first an adaptin protein and then also a cargo receptor. So only certain cargo proteins will bind to specific cargo receptors. So the clathrin and the adaptin will only bind to the correct cargo receptor, and that will help to select only a specific type of protein to be inserted into that vesicle. And so you can see that as that vesicle continues to form, that only one type of cargo gets picked up because of these specific cargo receptors. Now to actually bud off, we have a second protein called dynamin, which is kind of like a twist tie. It kind of tightens and tightens and tightens on the neck of that vesicle until it breaks off. Now you have a coated vesicle with the correct target protein inside of it. It doesn't really need the clathrin anymore, so the clathrin is released, right? So this process is used for both secretory and endocytic pathways, so exocytosis or endocytosis. In this specific example, we're looking at endocytosis, all right? But the same mechanism would happen, say, on the surface of the endoplasmic reticulum or of the Golgi. Again, this clathrin kind of promotes this basket type shape, and also important to know is the dynamin. All right, so the clathrin helps promote the shape, the budding of the vesicle. It also directs what cargo is in the vesicle. The dynamin helps to release that vesicle from the plasma membrane where it is formed. Once that vesicle is released from the bilayer, the protein coat is not needed anymore and it is shed. Again, how does this vesicle select its cargo? The clathrin is essential for this. The clathrin binds first to adaptin, and that helps to mediate the interaction between the clathrin and a very specific cargo receptor. So say we are making a vesicle of a neurotransmitter, right? So say serotonin, all right? So there is a specific serotonin cargo receptor. It only binds to serotonin, and that serotonin cargo receptor only binds to a particular adaptin, and that adaptin only binds to a particular clathrin. So that clathrin is directing, in this example, only serotonin to be put into this vesicle. Okay. So the clathrin, with the help of adaptin and the cargo receptor, determines what is in that vesicle. Now, how does the cargo receptor know which proteins need to be and leaving in a vesicle or not? So, for example, the serotonin, how does the cargo receptor identify that serotonin needs to leave as in a vesicle versus some other protein? These serotonin, or what other protein molecule it is that needs to leave the organelle in a vesicle, is going to have a specific signal on it that is read by the cargo receptor. So there are other types of coding proteins and adaptins. In addition to clathrin, there's also COP. There's various types of adaptins, and again, there's different combinations depending on where that vesicle is originating and where it is ending up. I don't expect you to memorize these, but just to know that clathrin is not the only one, and that adaptin, we have various types of adaptins. Now we have a vesicle that's been released, and it's traveling around, and it needs to get to its target destination, right? So vesicle docking, or kind of approaching its target and staying there, requires two things: tethers and snares, right? So you should know first, how do vesicles form? Next, how do vesicles identify their target location? So we have vesicle surface markers that indicate their origin and their cargo, right? So that is something that's really important for that target destination, right? So these surface markers tell the target organelle, for example, whether or not this vesicle needs to dock or not, right? So we have the surface marker called RAB, right? RAB is on the surface of a vesicle, and it interacts with the target destination via the tethering protein. So on the surface, we have a of the phospholipid bilayer, we have a tethering protein. Right? So here, for example, we have a vesicle that's going to be secreted by the cell. And so at the cell membrane, we have, facing the interior of the cell, a tethering protein. A vesicle approaches, and the tethering protein identifies that RAB. And if the RAB is the correct one, if the tethering protein reads it and says, yep, you're a vesicle that needs to fuse with the cell membrane, you are needing to secrete your contents outside of the cell. Now what happens after tethering is docking. So again, every organelle and vesicle has a unique combination of RAB proteins. So these tethering proteins will recognize the RAB and read, are you supposed to be here or not? Now we have, after tethering, we have docking, all right? So in addition to recognition via RAB and tethering proteins, we have snares. So snares, we have a V snare and a T snare, right? So this vesicle gets brought close due to the tethering protein, but it has not yet fused with the cell membrane. So V snare, vesicular snare, T snare, target snare, right? So they have to match. Only certain V snares will attach to certain T snares. So it's another method, another layer of recognition to make sure that this vesicle is really supposed to be here. Then it's pretty cool. These V and T snares will kind of twist around each other, and they will kind of act as like a winch, bringing that vesicle closer and closer and closer to the cell membrane until ultimately that vesicle fuses with the cell membrane. This vesicle has to come within 1.5 nanometers of the membrane, and what that does is that really forces the water out of that gap and allows those two phospholipid bilayers, so phospholipid bilayer of the vesicle and of that target membrane, to fuse with each other, intermix. And again, this is catalyzed by the snares, the V and the T snares. Let's stop into a problem. Right? So scientists are working with a mutant fly that at elevated temperatures becomes paralyzed. Now they know that the mutation is in the sequence for dynamin and it's affecting the structure of dynamin. And so the way this mutation works is that dynamin stops functioning at higher temperatures. So what function is being inhibited by this impaired protein that would lead to paralysis? So take a minute, pause the video, and think about this problem. Now we know that dynamin is important for vesicle budding, and we know that vesicle budding is used for the formation of vesicles for things like endocytosis or exocytosis. Now, how does paralysis, how does motor function work? We're talking about muscles and neurons, right? Now, if you think about it, in the last chapter, we talked about how neurons signal, and we talked about neurotransmitters being released at the synapse. They're waiting inside the cell, right at the surface of the cell membrane, right under the surface of the cell, in vesicles. And so these vesicles need to be able to form properly, and they also need to be able to exocytose properly, to be released into the synaptic cleft. So for these vesicles to initially form, we have to have vesicle budding, right? And we talked about how vesicle budding involves dynamin. So say again serotonin, or let's say acetylcholine, right? Say acetylcholine gets made. It's going to be made by a ribosome. Where is that ribosome going to be found? Is it going to be in the cytosol, or is it going to be on the surface of the ER? Well, we know that this acetylcholine is going to be secreted, so that means that it's going to be made by a ribosome on the surface of the ER. We know that that acetylcholine is going to have a signal sequence that allows it to get translocated as it's being made into the lumen of the ER. And then the ER is going to bud off, and in that vesicle that buds off is going to be serotonin. And the last step to release that vesicle so that it can travel down the axon to the synapse is for it to be clipped off with that dynamin protein. Right? So if there's no dynamin function, there's going to be no neurotransmitters ready to go in vesicles at the synapse, and no neurotransmitters are going to be released into the synaptic cleft, and muscles are not going to be activated. All right, so that is how misfunction of dynamin can lead to something as drastic as paralysis. Last thing that we're going to do, I believe, is just a good activity to help you keep track of the different transport mechanisms. So the two main mechanisms that we've talked about are signal sequences, right? So these peptide sequences that direct a protein to a target, and the protein travels through a translocator. And then the second transport mechanism we've talked about is vesicular transport. So we have vesicle budding, we have tethering proteins, we have T and V snares, right? So if a protein is going to, let's start with the endoplasmic reticulum. If it's going to end up in the endoplasmic reticulum, what signal transport mechanism is going to be used? Right? We talked about translocators being involved, right, helping that ribosome as the protein being made by the ribosome. It takes that protein and threads it into the ER. So that would be signal sequence. We talked about proteins that are going to be secreted. After they're made in the ER, they have to go through where? The Golgi, right? And so they are going to leave the endoplasmic reticulum via vesicles and travel to the Golgi. So this is part of that endomembrane system. And then if that protein is destined to go outside of the cell, it is going to bud off in a vesicle. It's going to be contained in a vesicle that buds off of the Golgi and then travels to the surface of the cell. So we still have nucleus left and mitochondria, right? So with the nucleus, we don't have translocators. We have a nuclear pore, but there is a signal sequence involved, right? We talked about this protein that hangs out outside the nucleus near the pores. If it identifies a protein with the correct signal sequence, it will bind to that protein and guide it through the nuclear pore. So a protein whose target is the nucleus is going to use a signal sequence transport mechanism. And then lastly, for the mitochondria, we talked about how the mitochondria has a translocator on both the outer and the inner phospholipid bilayer. And so if we're using translocators, that is going to be a signal sequence transport mechanism. Right, so again, Golgi, the proteins are going to be traveling to the Golgi via vesicles. For targeting to the nucleus, signal sequences are crucial. If in this one, I have it selected as vesicles, but really that should be corrected to signal sequences, right? If it's going to the endoplasmic reticulum, it's using a signal sequence. However, if it's leaving the endoplasmic reticulum, it's leaving via vesicle, so that's why I have vesicle here. Mitochondria, if a protein is destined for the mitochondria, it's going to be using signal sequences to be transported into that organelle. And then if a protein is destined outside of the cell, it's going to travel via vesicles to the cell surface. All right. So don't forget that there is an activity that you should get started on so that you can respond to my survey. And again, just pick out some top 3 problems that you want to go over, and I will discuss those before the assignment is due. Have a great day.